Welcome to Flow Stars, candid conversations between Dr. Peter O'Toole and the big hitters of flow cytometry, brought to you by Beckman Coulter at Bitesize Bio.
Welcome to Flow Stars, our series of interviews with top names in flow cytometry. We have another double header for you today as I chat to Jonni Moore of the University of Pennsylvania and Rachel Errington of Cardiff University. In this episode, we'll get some career advice from the President and President-elect of ISAC.
Your career pathway is a marathon, not a sprint. And I think you need to weave your way through it and make sure you look after what's important along the way.
We discover why Jonni and her husband and dog dress up as Victorians every December.
What they do is they close off the street and they put bales of hay, and various people sit there reading Dickens.
And discover how Rachel learnt the hard way about the role of lab pranks.
I realised then that you had to survive in this lab. You really had to raise your game in terms of the pranks that you had to play.
All in this episode of Flow Stars. Hi, I'm Peter O'Toole, and today I'm joined by Jonni Moore from the University of Pennsylvania and with Rachel Errington from the University of Cardiff, who actually have quite a lot in common, even though they might not realise this yet. I look at the horror on their faces when I say that. We have the current president of ISAC and the incoming president of ISAC. So it'll be great to hear what Rachel thinks Jonni's doing wrong and what she'll improve in the future. Also, they've both had a career in the cytometry with cell cytometry as a whole, not necessarily flow cytometry on the way through. And also both have startup companies, and actually I'd say more than started up companies that have been successful. So quite interesting careers and tracks to go through today. So Jonni, hello. Rachel, hello.
Hi, Peter.
Hi, Peter.
When did you two first meet?
Well, I first met Rachel years ago, but through actually ISAC, and when she became involved early on as not that long ago as treasurer and involved, and that is when I first met Rachel. During that time, I knew of her for quite a while, but that's when I first met her.
I was going to say there's two phases to knowing Jonni. There's knowing of her and seeing her in the distance and watching her operate, and then there's there's participating with Jonni and doing things, and all of that has been centered around ISAC.
I was waiting for one of you to have a completely different memory of when you first met.
No, I think the key thing here is that we have met a lot virtually, because that's how the society gets together most of the time, is by virtual, because we're international. And so, it's strange not meeting somebody this year because we've been virtual because of pandemic. So you kind of miss that once a year meeting, because it is the only time that you meet.
I agree. And I always say that I don't know everyone, but I know everyone who does know everyone. So in the cytometry community, in the greater cytometry community, it is actually a very close and interactive community. So you do know the names, you do. And the best part about it to me is that if I didn't know Rachel, and let's say I had questions about track 5, which I did years ago when I first started using some of that, I wouldn't feel upset about going to Rachel and saying, who's the best person to do that? Even though maybe I hadn't met her at that point. And to me, that is what is very unique about this community. And that is some of our challenges right now that we can talk about in a little bit, is keeping that close association. As Rachel said, we mostly exist that way, but we've always had a live face-to-face meeting where we can get together. And one of the main memories in my life is when we had our meeting, the first in Montpellier, France, many years ago, we had 2 ISACs there. But we used to leave the meeting, and then the cafes in that town would be filled with cytometrists, and we'd be sitting there drinking wine, eating good food, still talking cytometry. And I think that's what is unique about us.
Yeah. Of course, so it's interesting having the two of you here, because you also contrast each other. Because a lot of people who join ISAC see it very much as flow cytometry, as a society, and it's not. It's cytometry, the measurement of the cell. So if I was to ask you, Jonni, what was your first cytometer that you ever used?
An orthospectrum 3.
Okay, and if I was to ask you, Rachel?
So for me, a cytometer is a tool in which you measure a cell. And the first cytometer I used was a Biorad MLC 500 confocal microscope.
So truly cytometers, but I bet, Rachel, most people wouldn't think of the confocal microscope as a cytometer. They would very much see it as a confocal microscope.
Yep.
Jonni, absolutely, you're in a flow cytometer. And I think you've got a challenge here. The society has a great potential to grow even further. And it's getting that microscopy community in or aligned with it that is still, it's still very heavily flow cytometry biased, I would say, as a society.
It is, and we talk constantly about a challenge, and one is because the microscopy societies are pretty strong, and they meet the needs of that community. I would suggest that confocal microscopy really started bringing them close together, because we're using fluorochromes, we're using different approaches than we did, even though we did use those in regular fluorescence microscopy. I think confocal made it more like, in people's minds, what we thought about cytometry. But I think what we are going into an area where we're starting to think about this field as single cell analysis, not flow cytometry. We're certainly doing single cell genomics in our field. Most of our labs are doing that as well. So if we think about that way, and we think about even the journal, which is a journal of quantitative cell analysis basically now, then I think we'll come together, because each technology brings something unique to the field, and they're complementary. They're not one or the other anymore.
Let me throw this at you and maybe get Rachel to think about this one, because there isn't actually a natural home yet, because it's so early in the technology of spatial genomics and spatial transcriptomics and even spatial metabolomics. Mm-hmm. They fall between stools. The microscopy community are very much on their super resolution and volume electron microscopy, run the slide scanning type technology. I guess the pathology side it would fit into, but that's not really necessarily the technologists, and our user base wouldn't align themselves with that. Mm-hmm. And yet cytometers, again, it's now just a pure imaging platform. So is there a home and something we could do here, Rachel, do you think?
Well, so for me, the spatial aspect of being able to measure a cell has been fundamental to what I have been doing, and also the temporal issues of how you can measure a cell over time, X, Y, Z, and time. And I think there is a lot of crosstalk within all of this, because you're using a fluorochrome, so you have to understand how you can get a quantitative measure out of a fluorescent signal and a readout, and also the spatial aspects. So how, when you see a response of a cell within a tissue, how is the neighborhood behaving? What is the proximity? What is the context of that behavior, that functionality? And that's where microscopy has, and the capacity to image has its real value. So that's when you can think about the spatial aspects of transcriptomics and really understanding the neighborhood, the niche, the microenvironment, if you like, of the system. And I think that's so important because being able to break up a population is one thing, but how is that really assembled within the tissue? And going between all of those platforms and that capacity is the most important thing. So it is not a one size fits all. You have to be able to go across the platforms and really to understand the biological system at whatever level, whether that is at the whole cell level or at the transcriptomic level. The spatial aspect is important, the temporal aspects are important, but you need numbers and be able to have populations as well. If you have all of that, that's when you start integrating your data together, integrating the knowledge that comes out of that, and that's when you start understanding the system. So I think that's where we need cytometry and microscopy and genomics to come together and say, how are we really going to solve? This is the big challenge for us. We can sit in our silos, but that isn't going to work. We have to come together and really share how we interrogate the data from all aspects. And you need your physicists there. You need your mathematicians as well. You know, they all have to be involved in order to dissect that information out, because you can't do it on your own. And we're getting very close to be able to do it all now, and it's how we bring that together. And I think that's the future of cell-based measurements, if you like, for me going forward.
I think it's tantalizing and yet so frustrating, because all these things that are connecting together, actually really hard to connect them all together properly. My PhD student at the moment, Laura, and she is a top math student that graduated out of York, and we grabbed her because it is the maths and the computer science that we need to analyze the data. So she's using flow cytometry, but actually a lot of her data is single cell tracking and looking at the changes. And so she's using all these technologies coming together. And you know, we can teach her how to use a flow cytometer, how to image. Very hard to teach someone like myself how to do the maths and the computer science behind it.
So I've had lots of conversations with youngsters thinking about where to go in terms of their biology, and you know, both as undergraduate, graduate, and postdoc. And really, maths is going to be fundamental to that. And how do we upskill to data analysis and big data analysis? All these things that get fly around, but really our biologists need to engage in that. And so maths is the new black for biology. It's it's where we're going to make inroads so that we can integrate data, we can then interpret data, we can use modeling, visualization, and bring all of that together. So mathematicians are really important to us going forward.
And I think, Jonni, you'll agree, I'm sure, that high-dimensional flow cytometry analysis has taken off in a big way in the past few years, and yet still has so far to go.
Yes, so I think, and I guess, what I like to think about is there's a word that was floating around, and Beckman Coulter and Paul Robinson used it years ago, called cytomics. And cytomics really is where we are now in this field. It is not cytometry alone. It's really the integration of what you're talking about. And we recently just changed our facility name to Penn Cytomics because we're doing all of these things. Interestingly, I just hired a new postdoc, PhD in applied mathematics, as Rachel said. He's already created 2 or 3 new algorithms, but he's not constrained about this is the way we analyze flow cytometry data or whatever. Because he has no background in that. He's learning it. Several years ago, when we first started our company, one of my partners who formed it is a physicist, Wade Rogers, and he actually did a lot in computation. He worked originally in genomics, but he is someone who really learned a lot of the biology, but after the fact. So he thinks about it like a physicist. He doesn't think about it like a biologist. But I might disagree a little bit. And, yes, I'm old school, but you mentioned, how hard it's put it all together. I don't need to understand in depth all of those mathematical approaches. I need to find people who do to work on the team. And I think now it's really all about single cell measurements, single cells in different cytomes, because if you think about the cytomics, you've got the tumor microenvironment, as Rachel was talking about, as well as the disease, and the ability to integrate all this, because now we have the computational power to do that. And we just started another collaboration with a really big data guy here at Penn. He has the largest basically genomics database of neurodegenerative diseases in the world. He does it for NIH. And he came to me and said he was very interested in cytometry. He thought it was very cool. Can we do for cytometry what was done for genomics? And I think you're going to see a lot more of that going forward.
Which is good, because
it's exciting.
If you'd have looked 10 years ago, I think some of the flow cytometrists would have been looking around thinking, have we got a future? Where is it going? And it has really blossomed. It's really exciting. And I still hear peers around saying, oh yes, but we'll replace it with single cell genomics without a cytometer, and we can do all the imaging mass spec stuff, so we don't need the fluorescent fluorochromes. I don't see it. I think we're the starting point. And I think we're the ones that are going to be feeding these other technologies, because those other technologies would almost run out of steam, possibly, if we weren't getting that visual context and the single cell higher plex data. And it's that I think is going to push into
And there is a practical issue. So for 20 years plus of my career, I also directed the clinical diagnostic flow cytometry laboratory here at the University of Pennsylvania. So I did the research part and the clinical. And it's a unique perspective, because I always say on the research part, we see everything we can do. On the clinical part, knowing what can translate and think about what's paid for and all those things, everybody gets all excited about doing 10 colors in a clinical lab. I'm doing 40 colors on the spectral instruments plus, and we've got Cytof is doing many more. So the trick is getting these technologies, and that's something that's a passion of mine, that can directly affect patient care and patient well-being. And that is finding a way to translate from one to the other. And I think that's where we're going to be challenged a lot. We've got a lot of things we could do, but in the clinical world, we haven't changed much in the past 25 years.
So that was going to be my question, Jonni, is where are the converters? How do you convert what we do in the lab, accelerate that conversion so it is affecting the patient? We work very closely with clinicians at Cardiff, and we value that interaction, and we get samples coming from the clinics, and so that we can enhance our science. But I'm not sure that we really are impacting back the other way as fast or as we would like. And so it's really closing that loop down, making it narrow, and finding where the converters are. So you can be, I see an ecosystem full of pioneers, full of innovators and converters. And with the, and I haven't mentioned whether you're a scientist or whether you're a nurse or you're a clinician. It's those three kinds of individuals that really allow us to work together and impact on the patient. Because if we don't do that, we are being truly academic in the truest sense of the definition of academic.
Nothing wrong with that.
Really good science and 4-star papers. Apparently, that's what we get metric on. But ultimately, you want to have an impact on the patient. If you're working in the medical school like myself, that's why I'm there. I want to be having an impact on the patient.
I think one of the ways that I've always approached it, and I've done it in a couple of fields. Right now I'm doing it in the whole high parameter field. And, how do I translate that? I work with if you will, diagnostic. We do the clinical trials for the Parker Institute for Cancer Immunotherapy, which is a consortium of several cancer centers in the United States. But what you have to do is have the conversations with the clinicians. And I'll always remember the one that was the basis for us starting our company was with a cardiologist who came to me and said, if a patient comes to me and says, Doc, will I have a heart attack? And am I going to have a heart attack? He said, I can't answer that question. There is no test that I can do that can tell me. I can tell his risk is up there some. I can look at something like troponins if he comes to the ER and is going to have a heart attack in the next 3 hours, but nothing else. And the cardiologist, who we always joke that cardiologists don't know from a flow cytometer, it's not used in their practice. He said, I just heard about flow cytometry. What can we do? And that was the start of us developing a clinical-based assay to look at extracellular vesicles for risk stratification for cardiac patients. So that, again, I wouldn't have necessarily thought about that if I didn't have that interaction with a clinician who's telling me a problem and a challenge that they have. And I think we need to have that back and forth a lot, because what we have expertise in is the technology aspect. In our lab, we have both clinical and research, but in general, we're techno geeks a lot. That's what our field is a lot. So we can find applications to answer these questions, but we need a close partnership with clinical people to keep this going.
I think you brought up an interesting point at the end there, Joni. You are both technologists, and just changing tack a little bit, you've both been involved in spinning out companies to solve different problems. So in your case, you got Cytovas, Joni, and obviously Biostatus, Rachel. How you're in academic roles, you're developing a career that you're being paid to do, and you've got an idea for these spin-out companies. That spin-out takes a lot of time, effort, energy. How did you do that? Would you advise anyone doing it? What tips and tricks would you give them?
Well, Rachel, my approach was a little different. So
I was not a founder of Biostatus. So I arrived as a postdoc in the lab and became involved in Biostatus. And what I see is essential for an SME to be able to deliver, and what I think we're delivering by developing our fluorophores, is that it allows us to really understand what the common problems are and to see whether we can provide some solutions. So Biostatus is not providing the drugs, etc., but these are molecules that have to behave like a drug. If it's got to get into a cell, got all the same problems. It's got to penetrate tissue, cells, get in, label. There's a target involved. So it is a sort of drug discovery in its essence, a pro-discovery program, and that's the other side of the coin is looking at doing drug discovery. But I think in order to, it's a bit like, how do you share your science? You can share your science with publications. You can share your science by sending a bit of the molecule in the post, etc. But that's not going to have the impact that you want. If you want to have real impact in terms of people using your technologies, you have to set up a platform to do that. And it isn't going to be through really good publications, etc. That gets you the recognition and the understanding of what can happen. And if you want quality control and you want everybody to be using a reproducible method, you have to have a platform in which to do it. That costs money to set up. These things don't fall into the lap. So by setting up a spin out, what's the motivation behind that is that everybody has access to that technology. We're using it in a reproducible way. We're delivering something that we know is going to work in the hands of in everybody's hands. And that's indeed doing science. We want everybody to be able to use that fluorophore stroke product stroke assay so that all our assays are reproducible and can be, and so when somebody's using that particular fluorophore, we understand what it means. So it's a means of getting a technology into the hands of many in a very systematic way. So for me, it's a no-brainer. That's how you have to do it, because I can't think of any other way of providing that systematic approach. Now, in terms of effort, you have to, and separation and conflicts, you have to be sure that you understand what those are. And the key for us at Biostatus is that the business is run by a non-scientist, a talented businessman in his own right and knows how to run a company. And us academics are involved in that company and steer the product development or the innovation. But ultimately, it's the businessman that keeps the whole show on the road. And so again, it's a partnership. It's like anything. It's a really good team that you put together and you partner with to make that successful.
I think you may have missed one other key aspect there from a customer's perspective is the support, the help and support that is always available. And in an academic post, you can't be doing that. You have your own priorities in an academic post, whereas the company gives you that ability, which is good. What about yourself, Jonni?
So, first of all, I call myself an academic entrepreneur. So I have always stayed in the academic environment, although I mentioned my partner, Dr. Wade Rogers, he did leave Penn to go full-time to Cytometry. We at Penn have rather stringent controls put on us. So, for instance, I cannot have any named role in the company, which is a stupid thing because if you don't let the inventors have a named role, it comes out. I am an owner, and Penn mostly owns it, but I can't have anything to do with, because I can't even talk to investors if they're raising money. So it's very much a challenge. But what universities don't do well, we do very well in research of the R&D paradigm. We do the research very well. The development to get something that can be out to the world, as we were just talking about, because that takes a different kind of money. It's not the money you're going to get from a funding organization in general, because it's developing the process, and getting that out requires raising money. So on the other hand, what's been very exciting about doing this is I can see a way, since my goal, our company was focused on developing a diagnostic prognostic assay, a system that can be applied to predict cardiovascular disease at first and then now traumatic brain injury, that could go out in the clinical world. And how's the best way to deliver this? We don't want, early on we worked on a platform that was the only one of its kind. And the company that developed it didn't have any plans to develop another one. So that's no good to us. I mean, it was good to us when we're developing the assay, but what good is that? Because you need to have these things out. We want to have them out. But I think the synergy that I've been able to do between developing those assays, again, talking to the clinicians, what is your challenge? Using the standardization, which is very critical that you do in a corporate environment. Not that we shouldn't do our research level that way, but it's much more stringent in that world so that you could develop. It's been exciting. And as far as Cytometry goes, it was, its next generation is in the process of developing. The first generation basically was sold and sold to the American Heart Association. They bought it, all the IP. And because they have an entrepreneurial side, because they want to further develop that. And now we're working with some new investors and a new around the traumatic brain injury and some solid tumor liquid biopsies versions. But one part of me says, if I'd started this when I was 20 years younger, would I have gone on that path? But then I think about it and I said, that was able to happen because I was where I was. I was in the academic environment, able to bring that knowledge and move that way. So I think it's very exciting. I think it's really, I've had the opportunity in my career to see from the beginning. I started out in basic science. I mean, I used a little bit of flow cytometry, but I was really focused on basic science. My science became more technology driven as I went along, and then to the far end on a commercial platform. So I think it when you can take it to that breadth, I think it's very exciting time. Has been for me, I know.
So, but all that takes time and effort, and
yes,
neither of you look completely exhausted. So
it's Monday. We had a weekend.
Yes, you do have outside interests. So actually, I've got this picture up. Jay, explain this picture.
So we have my husband and I, that's my husband, Roger, and my baby dog, Willow. We have a house on the eastern shore of the U.S. on the Chesapeake Bay, and there's a small town that's called Chestertown. And the first weekend of December, they have a Dickens Festival. And what they do is they close off the street, and they put bales of hay, and various people sit there reading Dickens. And then they have, English food, and it's really great. So my husband and I, everybody does dress up. We got into the spirit of things. So it's great fun.
I'm intrigued to know what English food is.
Yeah, tell us what English food is.
Well, what they're doing are those, the hard-boiled eggs with the sausage around them.
Oh, yeah, Scotch eggs, yeah.
Scotch egg. So it's generically English, and there's some, I guess it does have a penchant for Scotch because they have haggis, and they have lots of things of those types of various sausages. And but they also have, since we're on the Chesapeake Bay, fresh shucked Chesapeake oysters that we do it. But it's great fun. We love doing that and dressing up and listening to all sorts of Dickens.
What's your favorite Dickens novel?
Well, Great Expectations is still my favorite. And I remember the first time I read it in high school, was that thick and going, I hated it. But then I read it again in college. I mean, I didn't hate the novel. I hated the fact that it was so long, and in high school, I didn't want to spend all that much time reading things. But in college, I read it again, and that's really one that's up there. What about you, Rachel? What's your favorite?
I too like Great Expectations, and Pip is one of my favorite characters. And actually, if you want to see a really good film, the one with Richard Burton in it, I can't remember when that was. I mean, it's a black and white film of Great Expectations. It is brilliant. It's really a great one of the great classics. A Sunday afternoon in the rain when it's raining outside.
Yes, we had a lot of that recently.
I think it's 1950s. Great Expectations is a great
Alec
film.
Guinness as well, isn't it?
Yes.
As it comes up, so it's on the salt marshes, isn't it? The opening scene is that murky, that really atmospheric, and I spent 10 years in Brightlingsea with salt marshes on the doorstep. Loved it for that reason. But the opening sequence for that was awesome. But here's a quick question, Rachel. What would you prefer, a film or a book?
Well, I would probably say a book, but at the moment, can I change the options? Because at the moment, my best friend is the radio, because you only have to use your ears. You don't have to watch. You don't have to see a screen, because, as you know, we're all screened out at the moment. So the radio has become a really good friend of mine, and it's amazing. It's got real depth of humour and intelligence, and up-to-date stuff, as well as storytelling. So the radio has really become a big part of my life at the moment.
And what about yourself, Joni?
I would say probably film. And part of it is because a great sense of guilt, in that if I take time to read something that's not science, I tend to be guilty because I can never read all the science I should read. And that's something I've never been able to overcome. I don't read 24/7, but I feel guilty if I don't. Somehow I don't feel guilty if I'm watching a film. It's, it's officially that.
Okay, so next one, film or TV?
Film.
Film?
Definitely.
Real life film I like best, but nowadays I end up watching, and especially after having gone through all this where there was no film to go to, but I virtually never watch TV shows. I only watch film on TV.
Yeah, I don't watch TV. I watch films on TV. I don't watch TV.
Yeah. Same here.
You got to have a bit of trash every now and then just to zone out.
Hey, look, a dirty secret I'll reveal is that when I just want to chill and know that it's going to be a feel good, I watch Hallmark movies. So we know how they're all going to end. It's all going to be happy. The right person will end up with the right person, at least the way you think. So that's my guilty pleasure.
I think we need that at the moment, don't we? That sort of prediction, because at the moment it's so unknown. So something that you know where the ending is going to be, I think, is perfect.
So, what are your favourite films, Rachel?
So my favourite film from when I remember being the first film I felt really bowled over by it was Gandhi. I remember being a teenager and thinking this film has really got me. I am forced to watch Star Wars movies by all the boys in our family, and they've really grown on me because I'm being educated about them. But I love a great film I love great filmmakers and that sort of impactful sort of. Gandhi was like that. It was a huge film. It was a film of scale, as well as talking about the individual. So I think I still stick to that film as my favourite film because I remember the feeling of coming out of that film, and I think that's when you know that it's really got you.
And Joni?
So I there's three films that I really like for different reasons, but I particularly like films that are very reflective of the times they're in, that have other aspects. I mean, they're a good story, but they have other aspects. And probably, I grew up in the South of the United States, so I have a perspective. So Gone with the Wind remains. It's a huge commentary on the time, and there's everything from strong women to comments on slavery, the economic differences between the North and the South, and not everybody looks at those. It's a very long movie, so they'll look at the love story, but I could watch that all the time. The other is one of my all-time favorites, is To Kill a Mockingbird, and the original one. And I have not, just before everything went kind of haywire, there was a Broadway play that was made of that. And my daughter, I think Jeff Daniels played the lead in that, but my daughter saw it and thought it was phenomenal. Of course, she had never seen Gregory Peck in that movie, so she went back and saw that, and really good. And I will agree with you, Star Wars. My son is a fanatic for Star Wars. He was born in 1980, close around, I mean, he didn't see the first ones because he was way too young, and ultimately did. And I have really grown to like some of the stories. There are some of them that I think are very commercial and aren't keeping with the stories. But I think George Lucas is a great filmmaker, or was. I don't know, I'd say he still is, but he was at a time. So those are the really ones that I really like.
So To Kill a Mockingbird was also coming to London, and myself and my two sisters were going to go and see it, and of course the pandemic stopped that. So I guess it was coming from Broadway,
yes,
to the West End, and we were going to go and see it. So To Kill a Mockingbird is definitely
so
good.
hopefully we will all have a chance to see these things sometimes again.
So Peter, thank you for this experience, because I now realise I've got more in common with Joni other than cytometry. Like the same films.
God, moving through. Rachel, this is something you sent through to me. So what is this picture of?
This is my tribe. This is fundamentally my group of my people, as I call it. So I have two sisters. They're standing behind me. I'm in the middle, and their children and our husbands. And this is really the group that really, when I'm feeling low, feeling high, feeling anything, this is the people I go to share. We're sitting on a log, which is a big piece of oak, and in the New Forest. And as you know, New Forest oak was used to build the Mary Rose, which is a Henry VIII ship that got sunk in the Solent and fighting the French and all of that. And so my mother is from the New Forest, and we were there to celebrate her life. But more importantly, the Mary Rose is a project that's in the family. So my father was part of raising the Mary Rose. He was part of the that supplied the compressed air, that supplied the air to raise the Mary Rose. If you remember it was coming up.
That was a big thing in the UK, wasn't it?
It was a big thing. It's a big thing. And so it's a very special place for us, the New Forest. And so we were there to, so I think it says, for me, it says, because it's all about context. It's like all images. It's not just the people in it. It's where you've taken it. And that has a lot of context for me in terms of we're sitting on a piece of oak that was part of the supply for building the Mary Rose. And so that sort of links it all together for me.
Oh, that's super cool. That's really nice. So looking on the light, we've talked about some quite serious stuff so far.
Yeah, let's lighten it up.
Let's get back into the lab. So actually, I'm going to put it. I don't know which way I'm going. Maybe think now. I think it's really important. You both reached the pinnacle of your careers. You both come up for presidency, got presidency of ISAC. You're both in very high positions within your institutes, your universities at the moment. But it's never plain sailing. Can you think of what maybe is the most challenging moment of your career to date? I'll start with Rachel on that one.
So I think it's really taking advice and finding the right champions at the same time as ignoring other people's opinions. And in the end, you can only put a pathway together that is your pathway, because life hits you at the same time as building your career. So I think it's really keeping true to yourself, and sometimes that's difficult, and really trying to being sharp and thinking about things ahead, but also being able to adapt. And I think that's really important, and not feeling burdened by what should be happening, what possibly what others think should be happening to you at certain times, and really looking at your career pathway is a marathon, not a sprint. And I think you need to weave your way through it and make sure you look after what's important along the way. And for me, that has been sometimes stepping away from my science and thinking about elements of my family that's needed my attention and not feeling guilty about that, but also knowing that, I can weave these things back together again. And it's really taking the challenge of weaving life, outside life, with scientific life. I think now that I have a status of responsibility, it's really thinking about how we can change things and attitudes on the ground of saying it's okay to have other priorities. There isn't a 4-star paper. There isn't writing a grant. It is okay at times for you to not think that that's the most important thing, and that's not why you're coming to work. It's okay to feel like that. It's important to tell people that, that's okay to be able to communicate that. And I think if we can have that supportive environment, I think we will do much better. We will keep our talent, we will nurture our talent, and the talent will rise to the top. I suspect that's not always happening because we're not looking after all of the aspects of being a person doing science.
I think that's sage advice. Jonni, most challenging time?
So I think to have a particular time, I think Rachel alluded to it, it's perhaps initially being a woman in a technology field that didn't have very many women. You may not realize, but this time at ISAC, with me as president, Rachel as president-elect, Jessica Houston as treasurer, and Kylie Price as secretary, is the first time in the history of ISAC that all 4 officers have been women. And I am the first woman president in 15 years and only one of 4 total. So the process associated with that was very early on. I found it difficult to balance family life because I didn't feel like I had that freedom to do it. That if I wanted to be a success, I mentioned I had a son in graduate school my second year of graduate school. So I really struggled with that at the early times of not being there for things for them, for activities at school or various things of that sort.
So but do you feel guilty? Do you feel guilty?
Yeah, guilty. Absolutely guilty. You feel guilty in that you, it's not that you don't want to do that, but you early on didn't see that there was any other path because it was still looking in an institution where I am, in an Ivy League institution. If I looked up the ladder, I didn't see me. There was nobody that looked like me, either a woman or a woman who had a family and didn't hide it. I never hid it. But I found some very good champions along the way who, by the way, most of them happened to be men, but that's because there weren't too many women around to do that. But they were great. And you've overcome this when your dean tells you on a phone call in June, Jonni, I know you'll be able to handle this because you're a force to be reckoned with. So I and as I responded to him was, and don't you forget it. But the point is, I think that exactly what Rachel said, one of the things that all of us have a responsibility for is it doesn't have to be that way. To let our up-and-coming leaders and the young people entering the field and say, it's important. You need to come forward and say, I need to do this. I need this help. And I think, if anything, the lockdowns that we've had where everyone was forced to take time away realizes anything can happen anytime to anybody, and we can adjust. It doesn't mean your life is over, your professional life is over. And I think if we take anything away from what we've experienced in the past 6 months, that's what we should take away, and really work with people to have priorities in the right place. I had one tech who had a lot of family responsibilities, and we had a lot of 12-hour experiments. He'd frequently start at 8 p.m. But that was his choice to do that. It didn't matter. I mean, he could do it whenever he wanted, but being a lot more flexible, and that's what I've come in. Right now, the whole childcare impact because of virtual schooling that we're doing here is really impacting our postdocs, our techs, and our young faculty. They can't come back full time because they have to be home, not only to babysit their children, but to teach their children.
Even without COVID, is the system still right? How have we come on far?
No.
We're in a much better place. I mean, science is a team effort, right? Team science, right? This is what it's about. It's not about the individual.
100%.
So I think what you have to do is you have to, and as part of a team, you can compensate for strengths and weaknesses, and you can deliver. You can deliver really high quality science, impactful science in using a team. But the problem is that still the metrics and the rewards is based on the individual, right? And so those two things don't match up yet. It's getting better, but it's not there yet. You are promoted and metric based on your individual performance, but actually the multidisciplinarity that we talked about right at the start is team effort. And once you're working in a team, you can compensate for the fact that life happens to individuals, and you can build some redundancy into all of that system and deliver really high quality, excellent science, but working as a team. And I love working as a team. I mean, it's so much more fun than working as an individual, and sharing the glories and the upset when things don't work out how you think they should be working out. So I think team science for me is a solution to many things, but until you're recognized for working in a team and that teams are important, you're still going to always get those conflicts. And, if you're going to demand somebody to work 60, 65 hours a week in order, and because that is the norm of being a good scientist, you're going to lose real talent.
And I think you mentioned, I've heard you mention in the past, Rachel, about careers don't have to be at 100 miles an hour all the time. They can take a period of sort of slowing, so you can have some family life and then going back up again.
I think it makes you a better scientist as well. Because
I agree.
To be a good scientist, you have to be aware of the world, aware of the challenges, and you have to be also looking after your family and and when you get in a place of seniority, you're looking after other people's families as well. So I think it's about pacing. It's about understanding when you have to put that extra effort in, extra time, but it isn't extra. It's just part of what you're doing in order to get yourself and the science funded and delivered in a way that really works. I've been a caregiver for 30 years, and there were times when I had to step away. People rolling their eyes, saying, "Well, you forget it. You're never going to get to the next stage. You'll never be asked again to give a keynote at this meeting because you say you can't go to that meeting because you've got a family commitment."
And look where you are now.
And it's just like, well, tough beans, because I cannot come today or next week because I've got other priorities, and that's okay. You learn now that actually it's okay to make those decisions, and it's the right decisions, because I'm sitting here knowing I've made the right decisions along the way.
I think there's a good message for everyone there, no matter what gender or bias you are of anything, is to always understand from the other person's perspective and have that tolerance and understanding to enable people to balance their lives. And there can be big things going in people's personal lives, which you'll never be privy to. And you have to understand that could be happening. Always just don't knee jerk.
Yeah, and don't judge, right? Don't judge. Why is Peter not here this year? Why haven't I seen a grant from him? Why haven't I seen a paper? Just don't judge.
Absolutely.
Don't do it.
So thinking of teamwork, you also have an interest in football,
Yes.
if I'm correct. So this is
Brentford Football Club, the bees. Hence, you see the symbol of the bees. The bees. Come on, you bees. This is a West London football club. And it is the club that my father always supported, because the other thing that you learn is that your tribe does direct a lot of how you behave. And so I've always supported Brentford. I will always support Brentford, because the memories of sitting down, watching Brentford, or phoning my dad on a Sunday and saying, did you see the results from yesterday? You know, they did well, they did badly, etc. Is part of my DNA, is my conversations about Brentford. I've lived in Wales for 25 years, coming up to 25 years. And the Bees play Cardiff, and I know the Cardiff team really well. I mean, I'm into my football. I know my football, and I know the Cardiff team really well. But I cannot support Cardiff. I could go to every match, and I love football. I love watching football, but I can't go and support Cardiff. It would be the sensible thing to do, because they're on my doorstep. Because my connection is not just the football, but the people whom I'm connected to and watching it with. It's the same with Wales versus England. I know both teams really well, but I cannot put on a Welsh shirt. I love Wales. It's been part of my life for a very long time, but I can't put on a Welsh shirt, because England was the team that my father and I would always sit down and support. And so you just can't overcome that. And so that's the other tribal nature of supporting a team, is that.
So with a couple of good seasons, you've had quite a lot of phone calls, whereas with my dad, after every match, there's a quick call and go, let's not talk about it, and phone back down.
Well, that's the other thing, sort of very depressing. I mean, we've had a really good season, Brentford. We nearly got promoted. We bottled it, and that's very disappointing. But ultimately, they're my team, and so we're back to square one for the new season, and I'll support them all the way through it. Yeah.
It's more exciting that way. So from football, Joni, you also support what is supposedly a football. It's not real football, though, is it? Yeah, we've got to face it. American football is not football.
Well, we could argue about that. So the Philadelphia Eagles. So I grew up in Virginia. We did not have a football team. We had the Washington, now called the Washington football team, but they were called the Washington Redskins at the time, was the closest we had for Washington, D.C. But I was, in high school, a cheerleader for 5 years. So I was very much involved in sports. So really, I can relate a little bit to what Rachel said about the memory. So my parents were not sports fans. My husband and my son are definitely not sports fans. My daughter is a crazy sports fan. So she and I have gone to football games since she was in middle school. So we've gone to Eagles games, and we've been crazy Eagles fan and painted green stuff on her face and all of that. But it's a lot of memories of that. And yes, she could not have married someone who was not an Eagles football fan. So she did. And my son-in-law is equally a rabid Eagles fan, as are my 4-year-old and 6-year-old grandchildren. So we come any Sunday, you're going to hear us talking, yes, our team's not doing too well now. They lost the first 2, so we do, like Rachel said, commiserate. Oh, that looked pretty bad this time. But that is definitely something that is a unifier in a lot of ways, and it does bring you, the family memories and all. So, yep, go Eagles.
So the other thing for me to say is that I was a teenager in the States. I went to school in New Jersey, and girls in the States play soccer.
Yes.
So that's where I also got my bug for football, was the fact that I actually played. Instead of cheerleading the boys playing football, I would go and play soccer, and you could do really well. So I played in all the school teams. I came back to the UK where girls are not playing soccer or football. They are now, but they weren't then. And I played football when I came back, and played at Cardiff University as well. So, you know, so the United States, ironically, gave me the footballing skills to actually play football because I was a teenager in the States.
So Joni, Rachel, at a microscopy meeting, there is a pre-Congress football match, soccer match. I'll translate for you there, Joni. Soccer for football.
Yes.
Football, no.
The round version.
Yes. Where it's actually the core facilities managers, heads, take on the commercial groups and the commercial team. And it's very much mixed gender on the pitch. Has to be, it's all in good spirits. But you should bring that to ISAC, surely.
Well, if we have a face-to-face meeting in 2022, which we're hoping we'll have, which will be Philadelphia, would have been Philadelphia now, I could probably wangle the University of Pennsylvania's field, and that would be a great thing to do, I think. It would be lots of fun and to do that.
Yeah, I'm a winger.
And I will be the cheerleader.
We'll have to teach some of the Americans.
Rachel can play. Rachel can actually play, but I'll be the cheerleader.
It's a good icebreaker. I like the fact that it is from the facility side against the commercial side.
Yeah.
Because I think it breaks down there are perceptive barriers between the dark side, as some people would call it, and the academic side. Actually, there is no difference.
Not as much anymore. A lot of the commercial side have come from our backgrounds and just chosen a different career path where their skills are really needed. And we need the companies to have those skills inside them. So I think those informal networking things are terrific for both sides. And it is all companies come together. And even for them, some of them wouldn't talk together very often. And actually, it just brings walls down. It's really... Yeah. So there you go. There's an idea for something to work out in the future. Yeah. Thinking of light entertainment, can you think, well, it's always a revealing question. You can always think of some of the darkest moments in the lab, but usually I always think of some of the funniest moments I've encountered in the lab. So I'll start with you this time, Joni, because I give Rachel time to think on this one. What's been the funniest moment you've seen in the lab or in the workplace, whether it be conference or lab, wherever? Well, in the lab, so my lab group, I've been very lucky. Most of the people that work for me, my senior people, have been with me 15 plus years. I actually have 3 people who worked for me for about 10 years, left and went to biotech for 10 or 15 years, but came back in the past 4 years. So believe me, there is a pay differential, but they just like being here. So we always used to dress up for Halloween. So this is a group, not all of my lab. My lab decided to dress up 1 year for Halloween as me. So they all got blonde wigs of a sort, and it didn't matter their gender or race. They were all me, and I came into work, and they were all like that. There's another one that I didn't send you that there was from Becton Dickinson years ago. They had, when they developed an instrument that had, was a fax vantage, and it had Diva software, but it also had something called an automatic sampling system, ASS. So they all, again, dressed up like me, but then put a sign on their behind that said, Diva with an ass. And said, we just bought that. So so that was good. But on a national level, there was an ISAC in San Diego, not the most recent one, but years ago. And the funniest thing was several leading members of ISAC putting on a show as the Village People, doing YMCA, totally dressed up like the Village People. And that was really great. that's one of the things I absolutely love about this field is that people, we're not only colleagues, but we can be friends and we can interact. And maybe you only see these people once a year, if that. And it's very interactive. And one of the things you mentioned about commercial is the synergy that's always been between the commercial side and the academic side. I have never seen in any other discipline. My husband is a physician. The pharmaceutical companies don't have the same relationships with the physicians as we do. We need each other. We build each other. And I think having that kind of thing where, that's why I think the football game would be a great thing, and why we're trying to do more in that area. So that is, we're a community, and both sides of the community, and thinking about what's going to happen in ISAC going forward. That's something I think both Rachel and I are committed to, is involving our corporate partners at a lot of levels. We have many of them, several of them we've been asking to be on committees, and a lot more involved than we have. And because you can't live without us and we can't live without you.
Very much so. Rachel, what about you? Your funniest moment in the lab? Or,
Hang on a minute.
no, this is going to be interesting. Do you know what? Joni?
Yes.
Just disappear. When she comes back, we'll be gone.
Yes.
So I've just realised that. So I live by the sea. That's so funny, guys. I heard you say that. Right. So I live by the sea, and just before this afternoon, it's been a beautiful day, and I use my binoculars, and they are Zeiss binoculars, and I won these in my first microscopy meeting, which was Micro 90.
Oh, wow.
Hammersmith, right? That was my first microscopy meeting, my first paper that I gave. And I put my name, they said, sign the card, they say, sign the back of the card, put it in. And I got a message to say that I had actually won the binoculars, and they were on their way, right? And so I was waiting for them. And then they arrived, and our lab technician, Barry Martin, I remember him well, and he taught me lots of things about being in a lab. And he said, your parcel has arrived, Rachel. I think it might be the binoculars that you won. I thought, this is great. So I unwrapped them, and there's this great big box, and unwrapped them. And in there were 2 eyepieces from a microscope, which were sellotaped together, right? And I pulled them out, and I had unwrapped everything as if it was new. I mean, didn't look tampered with at all, this box. And I took them out, and I went, these are 2 eyepieces. I haven't won 2 eyepieces, have I? And the whole lab just fell around laughing, because of course they had completely unpacked it. And they went, no, this is what you won. So I realized then that you had to survive in this lab. You really had to raise your game in terms of the pranks that you had to play. And so that was my initiation to doing pranks really well.
Are there fewer pranks now in labs than there used to be?
Yeah, very boring. No, I think
there are far fewer pranks in labs.
There are far fewer.
Oh, yes.
I think
again, it's like we said earlier, the pressure to succeed, which we really, people trying to balance things.
Is it that, or is it the fear of doing wrong or upsetting or offending someone? I think
I was going to get to that as well. Yeah. And we live in an environment where people, I hate to use the often used term of political correctness, but I think because the problem with this is because there are really truly issues within academia and labs of inclusion and diversity, so that they're more, I won't say they're more important now than they used to be, but people are very aware of them now, for rightly so. So it's harder to not think of those things and to do the usual lighthearted jokes that we do. We still find time, and you know, the picture that you deleted is me in a witch hat. Again, I said Halloween. We still have our Halloween issues, and we still do it. And I had a big black mole on my nose and all of that. And I used to wear my witch hat whenever we would have lab meetings, and when I was going to be a witch, or another term that rhymes with that my lab would say. But I think, you know, we are, and plus right now, I think this whole epidemic is going to change things for a while, because it's almost like people feel like, well, life is so serious, there's so many problems happening, that we shouldn't be joking around so much. I mean, everything's so serious. And plus the fact with social distancing and all, I come in my lab, I've got 12 people in the lab, I don't see them. Everybody's in different places because they're separated or coming at different shifts.
Yeah, I think we got more serious before that, though.
I think you can have fun, though, can't you? I think you have to be
Yes,
careful.
absolutely.
You have to be mindful. You have to be aware of who's everybody around you, and etc. I mean, I'm starting to sound like a director now, but you can still have fun.
Yes.
And I think that's critical, and an essential part of doing science is that fun element. So, but you know, a prank here and there, I don't think would.
Part of being a team.
We are over the 1 hour mark somehow. So it's been brilliant. But I've got to ask just one last question. It's going to be a really short answer because I realise time is up. What's the next big challenge? What's the unmet need? What's the next development that's needed? That's a really long answer, I know, that could come out of this. But it's short, sharp. What do we need to solve next, and how? Rachel, with you first.
Yeah.
So I think it's pulling together the systems thinking on putting our insights together and how we really understand how the system works. So that's an intellectual way forward, and the technologies that underpin that are really about the science, about the maths and the engineering of systems and how the information is brought together there. So I think it's working from my perspective. It's working with our physical scientists to understand biology. I think intellectually that's what we have to be doing.
And Joni?
I would totally agree. I think we're at a step where we have tremendous technology. We've identified lots of needs. We've talked about, initially we called it personalized medicine. Now we talk about precision medicine. We can do precision medicine. We know how to do that. We can take what we have, but getting it from the lab to the patient, as we said before, is going to take this type of interaction, these teams with our physical scientists, our computational scientists, and that's what we have to do. We're not at a loss for having the technology. We've got it. We just need to know how to deploy it, and that's where I see our future.
I think you're both singing, so you've got a lot in common.
I know, exactly.
I've got a load more questions I wanted to ask, but it's gone all sorts of directions, which I think has been great. Joni, thank you. Rachel, thank you. You've been great to talk to. We'll have to do this again, and
Our
hopefully
pleasure.
at ISAC.
Yes.
Thank you.
Take care.
Thank you very much.
Bye.